Assalamualaikum warahmatullahi wabarakatuh, and good afternoon Dr. Marnim and my fellow friends. Today our group will be representing an article about the multiple roles of PAX2 in the embryonic mouse eye. Here are the beautiful members of group 1. First is Shakina, second is me, third is Nur Afika, fourth is Adina, and fifth Tang Yujing. In this research article presentation, we will cover about few contents. First is abstract, introduction, results, discussion, methodology, and conclusion.
Assalamualaikum warahmatullahi wabarakatuh, and good morning Dr. Marnim and my fellow friends. So today I'm going to explain about the abstract part of the article. So there are a few things that we have to understand in order to extract the important information from this article. So firstly, how does the vertebrate eye and unlarge grows? So unlarge are referring to the embryonic part of the eye which undergoes development, and this part grows out of the brain and folds into bilayer optic cups, and the eye is patterned along multiple axes, precisely controlled by genetic programs to portray the neuroretina, pigment epithelium, and also the optic stalk tissues. And secondly, we have to understand about the common role of the PAX2 gene. So the PAX2 genes encode developmental regulators of key morphogenetic events, with PAX2 being essential for interpreting inductive signals, including in the eye. And then we have to know, what if PAX2 gene is mutated? So PAX2 mutation can cause ocular coloboma. So what is ocular coloboma? It is a situation where the optic fissure in the eye fails to close. So whenever a person have this kind of disease during the embryonic eye development, the person will have a problem in order to view at a certain field of the eye. So in this article, the researchers are using few mutated mice, such as the PAX2 GFP plus knock-in allele, and when this mutation were induced in the mice, they observed that there are few defects happening, which prove to them that the absence or the lackings of PAX2 gene can actually cause multiple disease. So from there, we know that PAX2 gene have multiple roles in the eye development. So when the researchers conducted this article, they find something important, and these findings are very crucial for the next development of information in order to promote a better understanding and provide a molecular model about the ocular coloboma issues, especially in human.
Next, moving on to introduction. The mouse eye formation begins with specification of the embryonic eye field within the anterior neural plate, which is then followed by bilateral optic vesicle outgrowth from the ventral diencephalon. Next, the optic nerve head, or ONH, is an important structure in the developing eye because it is comprised of lineage-restricted cells that serve as a boundary between the neuroretina and glia optic stalk. Failed optic fissure closure, or coloboma, and abnormal ONH formation arise in variety of congenital diseases. A myriad of signaling pathways play essential roles in the coloboma network. Human PAX6 mutations are associated with multiple ocular syndromes, including MAC and Peters anomaly.
In this study, a mouse with a GFP knock-in that abolishes PAX2 function and expresses a eGFP reporter is used.
The first part of the results talks about PAX2 GFP plus reporter expression and the PAX2 GFP ocular phenotypes. Both of these are different results, so I will be explaining them in separate. So let's talk about the PAX2 GFP plus reporter expression. Why do we want to look at the PAX2 GFP plus reporter expression? This is so that we can confirm that PAX2 GFP plus reporter gene is a suitable marker for us to mark the location of PAX2. We have to properly confirm this to avoid mistakes when using the PAX2 GFP plus in future experiments. So before that, let us first see where PAX2 can be found. It can be found at optic vesicles, midbrain, hindbrain boundary, and developing kidneys. So now let's look at the results obtained by the PAX2 GFP plus reporter expression. So all of this is observed by multimodal imaging, which allows us to observe the GFP in the live mouse embryo. So what do these scientists observe under these live embryos? Firstly is the EGFP is visible within all of the PAX2 places. It means the aforementioned optic vesicles, midbrain, hindbrain boundary, and developing kidneys. EGFP is observed in all of these places. Okay, take note of that. And the second one is the EGFP is localized to the ventral optic cup along the optic fissure. Note that PAX2 also is going to be localized to the ventral optic cup along the optic fissure. So we can observe that the EGFP is also following the PAX2 where it should be. And lastly, EGFP is confined to the ONH and OS, which is the optic neuron head and the optic stalk, which is also the same as PAX2. And in the picture below, you can see that the location where GFP can be seen is also the same as where PAX2 can be seen. Both of them are the light colored parts there. And so, based on these observations, we can say that PAX2 GFP plus gene reporter is a reliable marker for PAX2. And now we can proceed to using this gene reporter in our next experiments. Let's take a look at the PAX2 GFP mutant inside of the mouse embryo, and we'll be comparing it to the PAX2 GFP plus mouse embryo that I've mentioned earlier. So then the first difference that we can see is the absence of PAX2 mRNA expression in the ONH and optic stalk. So in the PAX2 GFP plus, we can see normal PAX2 expression inside the ONH and optic stalk, which is shown by the picture B over here, this light purple thing. And however, in the PAX2 GFP mutant, we cannot see any PAX2 being expressed at the ONH and optic stalk, as you can see in this F picture over here. Next is displayed an enlarged GFP domain surrounding the normal position of the retinal ONH boundary. In the PAX2 GFP plus, where PAX2 is normally expressed, you can see that there's a boundary over here and the GFP is only a little bit over here. However, in G, where PAX2 is not expressed, there is enlarged GFP spilling into the retinal space over here, where the retinal ONH boundary is normal. Lastly is the lack of PAX2 mRNA expression in the hindbrain. So you can see in the picture A is the normal expression of PAX2, hence the normal size of the mouse brain over here. However, in E, where there is lack of PAX2 expression, you can see that the brain is there, but the size is significantly smaller than the PAX2 GFP plus. PAX2 GFP mutant has a smaller brain over here. And based on all of these factors, they concluded that PAX2 GFP produces null protein, which is non-functional protein for the mouse eye. Now, let's look at the outside development of the embryo between PAX2 GF plus, which is the row up here, and the PAX2 GFP, which is the row down below here. So, let's look at two things, which is the shape of the head and the development of the eye. In PAX2 GFP plus, in the picture A over here, you can see that the head has formed beautifully. However, in PAX2 GFP mutant, in the picture C over here, you can see that the head is caved in and has not developed beautifully like it had should be. And so next is the development of the eye, as seen in this A prime over here. The eye in the PAX2 GFP plus has developed beautifully. The fissure optic has closed perfectly. Although in C prime, in the PAX2 GFP mutant, you can see over here at the small arrow over here that the optic fissure has not closed properly. If the optic fissure has not closed properly, this will lead to future defects of the eye.
Hi, now let's move on to my part. Now you know that PAX2 is very important in the eye development, and there is an important step in the embryonic structure to develop the eyes, which is optic fissure closure. Optic fissure closure is basically very important. If the closure is still open, it would produce a mutated phenotype. Based on the article, they name several important transcription factors that involve in the eye development. Okay, some of them are RPC and MITF. So RPC is basically retinal progenitor cell. It is a specific protein that has a homeodomain. The homeodomain is namely VSC2, or we call it CHX10. So basically the homeodomain of the RPC is very important because it ensures correct timing and magnitude of the hedgehog protein signaling in the mouse eyes. We learned about hedgehog protein before, and in the mouse eye development, the hedgehog protein basically controls the cell proliferation. Cell proliferation means the cell rapid production or growth. And RPC is very closely related with the MITF. And MITF is another transcription factor in the eye development that is very important, and it is expressed by the NCC, neural crest-derived cells. So MITF is melanocyte-inducing transcription factors. So MITF is very important because it provides instructions to make a protein called melanocyte. Melanocyte is the middle layer of the eye, so this is during the eye development. That's why MITF is very important. So MITF is basically expressed by the NCC, neural crest cells, that is originate in the ectoderm at the margin of the neural tube. So neural means it is closely related to the nervous system. Okay, so the product, the MITF melanocyte that is produced would eventually enter or accumulate in the RPE, retinal pigmented epithelium. Okay, as you can see in the diagram 11.5, okay, this is reported expressed retina. And you can see the RPE is in monolayer, means it's still at the starting of the development. The fissure is still open. See what happens if one of the important elements are not expressed correctly. The elements mentioned here are VSX2, MITF, and FOXG1. For instance, because of course there are more transcription factors and proteins involved in the eye development, but let's focus on these three. So in the PAX2 mutant, the VSX2, it won't be expressed normally, meaning sometimes it could be truncated. So due to that, it will then producing abnormal eye development. So as you can see in the diagram E13.5, let's say we have a truncated VSX2 in the nasal cup. Basically, the VSX2 is shortened. Maybe it went through a deletion during its expression. It will cause the VSX2 to it cannot function well because it is shorter than its original size. For instance, the nasal tip could possibly be abnormally positioned, and in the diagram E13.5, as you can see in diagram C and G, it has a normal optic cup size, but the fissure closure is a failure. So now you know that the VSX2, MITF, and FOXG1 basically are important elements that control the PAX2.
Now I'm going to explain the discussion part of the article, and it composed of two parts. First is the PAX2 in the early eye gene hierarchy, and the second is the PAX2 in an integral component of CNS, central nervous system compartment boundaries. PAX2 expression in the eye field and nasal optic vesicle is governed by signaling pathways and CNS regionalizing transcription factors. One of the signaling pathway, SHH signaling, from the ventral neural tube, where neural tube serves as the embryonic brain in spinal cord. It establishes the DV, dorsal ventral axis, in the eye, and at the same time it activates PAX2, which means PAX2 depends on SHH activity, and its demonstration is the induction of larger PAX2 domain on optic stalk upon global SHH mRNA overexpression in zebrafish embryos. Besides that, PAX2 is also regulated by VAX genes, where in the absence of VAX1 or 2, SHH will induce this but does not maintain PAX2 expression. And PAX2 might also act during DV patterning. The optic vesicle or optic cup NT axis or nasotemporal axis is determined through SHH and FGF signaling pathway interactions, and these interactions result in the built-in expression of FOXG1 in nasal halves of the eye and FOXD1 in temporal halves of the eye. And we can see from the picture, nasal is the inner part and temporal is the outer part. FOXG1 simultaneously blocks WNT8B but stimulates PAX2 expression prior to retinal neurogenesis. In PAX2 mutant mice, nasal-specific morphologic deformity, or we can say its distortion, which could be the result of migration defects during ocular morphogenesis, or in other words, PAX2 is crucial for the morphogenesis of the eye. And after retinal, RPE, ONH, and optic stalk, all these territories are established, PAX2 activity remains essential for optic fissure closure and glial fate specification. Without PAX2, proximal optic cup and stalk fails to adopt a definitive tissue or cell identity. In the brain of an embryo, there is something called rhombencephalic isthmus. It is one of the boundaries in the developing CNS, which separates the embryonic midbrain from the hindbrain, which we can see from the pictures from the right.
and PAX2 is specifically expressed in the isthmus, and this is proved by mutant embryos exhibit brain excessively, where brain excessively is meant by malformation of the midbrain, isthmus, and hindbrain. One characteristic of CNS boundary cells is slower progression through cell division, and at the brain isthmus, high sustained HES1 expression is indicative of this deliberate pace, which proves this slow progression. And in the eye, the ONH also requires high sustained HES1 expression, which was not discernible or observed in the PAX2 mutants, PAX2 GFP eyes. So PAX2 maintains high HES1 expression at the slower division rate, and it is possible that PAX2 autonomously suppresses cells from converting to the faster division rate. In the developing kidney, PAX2 maintains a lineage boundary between nephron and renal interstitial cells by promoting nephrogenic fate, where interstitial cell means cell that lies in the spaces between the functional cells of a tissue. PAX2 null cells, that cells without PAX2, acquire the hybrid status of both nephron and interstitium progenitor cells, eventually differentiating erroneously as an interstitial cell, which means there's no PAX2, so there's no boundary between them. It forms a hybrid status. So the ability of PAX2 to recruit epigenetic modifiers of gene expression and chromatin structure keeps its kidney lineage on a particular developmental path. And in PAX2 mutant ONH cells are also unable to maintain their normal course, and eventually they will adopt the wrong fate.
So there are few methodologies that have been used in order to conduct the experiments in this research. So firstly, the researchers are using animals, which is the mice, and the mice were mutated in such a way that they want to understand the rules of the PAX2 genes. So the version of the mutated mice are the PAX2 GFP, plus mice, PAX2 GFP mice, PAX6 SAY mice, PAX6 CKO and CKO mice. And then we have also the OCT and SLO imaging, where the adult PAX2 GFP plus mice were live image and visualize GFP plus cells with retinal tissue context. And then the researcher also used immunohistochemistry in order to conduct the antibody labeling. And then there is RNA in situ hybridization, where the DIG level antisense riboprobes were synthesized from cDNA templates for GFP. And then there are also EDU incorporation, where the EDU pulse labeling was performed by injecting EDU in pregnant dams in order to assess the proliferations of cells. And then the researcher also conduct microscopy and statistical analysis, where some of the microscopes used are the Leica MZ12 dissecting microscope and the Zeiss Axio Imager M.2 microscope. Next, the researcher also used triplex RT-PCR, which is intended for the qualitative detection and differentiation of RNA, and the amplified RNA were used for cDNA synthesis. And then we have quantitative RT-PCR, where the PAX2 transcript were quantified by real-time quantitative PCR, and then it will then be used to compare samples to mRNA levels in wild-type optic cups. Now we have come to the end of the article. So in conclusion, this study reveals the multiple roles of PAX2 and PAX16 in the embryonic eye development in mouse, and these findings can actually help scientists to apply this knowledge in order to develop a better understanding of the eye diseases in humans. Knowing the causes of the disease, scientists can develop prevention and solutions to overcome the problems. Scientists can do gene knock-in or gene knockout such as the disruption of the gene in order to prevent the function of the gene, where the genes could be detrimental to the health of the patient, or gene knock-in, where we introduce a good gene to promote a better expression of a gene that is important for the eye development. So I think that's all from our group. Thank you.